Okay. Hello, my name is BJ Price. I teach biology at Twin Falls High School in Twin Falls, Idaho. We are looking at your Biology A review for semester one, and I'd like to start by welcoming you, and hopefully you can gather up as much information as we've gone over in 10 minutes. So here we go. The first unit that we started with was Unit 1, and that's Chapter 1 in our biology book, pages 2 through 24, and some of the main concepts. Simple and general starting out, what is the goal of science? And the goal of science is to explain the world and to make useful predictions. Just like gravity and simple concepts like that. We just want to make sure that we are understanding the world and what's going on around us. Being more specific about biology. Biology is the study of living things, and you can add that useful predictions about living things there. It's kind of neat how in the last few years we're starting to expand our knowledge and looking for life in other places. So I'm sure in our lifetimes biology and the definition will expand itself. Scientific method. So the next part is, how do we go about talking in science? And I kind of want to start talking about if you walked outside and your car wouldn't start, what would what are some of the things that you would do? And this kind of falls in the same thing as a scientific method. The first thing you would notice is that your car didn't start, and that's an observation. So scientists always start with observations, and from that observation, we and even you can take your car and go, okay, well, if my car's not starting, we start to go, what's the problem? And that what the problem question is becomes a hypothesis. So we're forming a hypothesis at this point. Why won't my car start? And let's say nothing's happening at all. The next part is, with that forming the hypothesis, we go, okay, I think my battery is dead. So what you would do in the next step of the scientific method is test that hypothesis. So we're going to test and see if your idea that the battery was dead is correct. So we would get some jumper cables and try to jump start. And we would then record the results. So we record the results. Did your car start or did it not start when you put the jumper cables on? And if it did start, you know that your conclusion, or you knew that your hypothesis was correct, and you can go to school or where it is you're headed. And if it didn't, then you'd have a different conclusion. Okay, why won't you'll have basically end up forming another hypothesis. So what are some other reasons why my car wouldn't start? So why that's useful to us and why we use this method is it's just a kind of a way to figure out things in a step-by-step process. And the next thing is, when we're applying this more to science and getting away from the car, we're looking at this as, how can I have someone else and even myself go back and repeat work over and over and over again, because what we tend to forget is that hypothesis is just a simple educated guess at this point. What we want it to be is something that becomes well proven, and once it becomes well proven, and a lot of people have done some replicating of your work and tested out that hypothesis many times, it becomes what we call a theory. A theory is a well proven hypothesis that no matter what happens, it always seems to be true. While there might be some things that they're still trying to find out about a theory, for the most part, the scientific community believes that as of that time is kind of, how do I say this, almost a fact, but not quite yet a fact. So a theory is kind of that step before it becomes a scientific fact, like the law of gravity. We tend to use theory in our everyday conversation as a way to sound smarter. In reality, we're actually trying to say what is hypothesis. So the next part, the tools that a scientist use. There are a couple things that I'd like to talk about. First of all, the simple one is a compound light microscope. Robert Hooke happened to be one of the first people that saw or found cells, and he found them on a compound light microscope. He actually had thin slices of cork made up, and he looked at them and saw that they looked kind of like monastery cells or gel cells. And so Robert Hooke was the first one using a compound light microscope that actually saw cells, a basic unit of life. There are other types now. There's a transmission electron microscope, the TEM. Basically that shoots images like an MRI through a microorganism so we can get a layered view of them. And then there's the scanning electron microscope. That would be like looking at the surface of a cell or a small organism. And it would look more like an orange peel image where you could see the surface of it. Along with that, scientists around the world use the metric system. I like to use it, one, because it's in multiples of 10. It's easier to do, to go from centimeters to millimeters and that. Some of the basics for the metric system are the basic unit of length is a meter, of mass is grams, and volume is liters. So just some things to remind yourself about as we're going through this review. The next part, we need to talk about controlled experiments. So there are two parts to a controlled experiment. A good experiment will only manipulate one variable or a single variable at a time. So the manipulated variable, as I talked about in class, is the thing that you messed with. And so in my mind, when you mess with something that starts with an M and manipulated, it begins with an M. So it's a thing that you're purposely changing in an experiment to see how it responds. Therefore, the responding variable would be what happens because you manipulated or messed with that experiment. So we looked at Reddy's experiment with the flies and the maggots laying their eggs, their larvae in the meat, and having larvae come from that. And what we did was we manipulated the meat by just covering it up with some gauze paper, and what we ended up seeing, the responding variable, was that there was no larvae anymore in that meat. Our control, on the other hand, because we left it open and the flies could get into it, it did have larvae. So our control was the piece that we left normal, what would normally happen. The one that we manipulated, the thing that we messed with, we will see the responding variable. So the next part of Unit 1, and again, I just want to reiterate this point. A difference between a hypothesis and a theory is a hypothesis is an educated guess. It's something that we come with a slight observation. A theory is well proven. There are so many works that have been replicated to make it a theory. So a theory, again, is the closest step you can get to a scientific law before it comes a law. The next part, the 8 characteristics of life, and we talked a lot about these. The first one are cells. Cells are the basic unit of life. New cells come from existing cells, and they form, when we talk about life in biology, we go by the definition that life has to, if they're not composed of cells, we automatically throw it out the window. So the 8 characteristics of life are kind of like the check marks, if you will, for how we classify life in biology. Another one that we look for is DNA. DNA, again, as we've gone through here in Unit 5, is replicating, and it gives all the blueprint information to how to create an organism. It's also kind of the history of that organism, if you will, kind of kept in the memory. So DNA is kind of like the memory system for life. The next one, the third check mark, is homeostasis. That is, does the organism maintain an internal balance? Every creature that we have looked at that we consider alive has some kind of internal balance system or regulatory system that keeps things even, or kind of at a homeostasis or a even balance. The next one is metabolism, and metabolism is just simply a way of saying how do we use energy. There are 2 different types of organisms that we predominantly talked about. Heterotrophic organisms, which have to go out and ingest their food, and then autotrophic organisms, which can make their food, like plants, using photosynthesis. Another one is growth and development. We just got done with Unit 5 again. The cell cycle is one of those growth and developmental keys that shows us what stages a person go through, or a cell, can go through. People tend to be born, they go through crawling stages, talking, learning how to walk, and as we get older, then we tend to go and have different developmental stages, adolescence, and then we end up going backwards and we get shorter because our discs are getting smaller and things like that. So even us have growth and developmental stages. One of my favorite ones was responding to the environment. Multiple times dropped books in here to make it really loud. We talked about having a cat chasing a laser. That's a response to the environment. Number 7 is evolution, and this is a change in a species over time. It's just not a change in a lifetime. That's growth and development. but evolution is a change in an entire population or a species over the course of a longer period of time. And the last one, number 8, is reproduction. And there's 2 types of ways, to keep it simple, for reproduction. One is asexual, and the other is sexual reproduction. And then the last part, and those were the 8 characteristics of life. The last part for Unit 1 is, what do biologists study? What levels do they study? And we started with atoms and the periodic table of elements, which should be hanging in every classroom. You got atoms, they come together to make molecules. Some of the important molecules for us are C6H12O6, which is glucose. H2O obviously is a pretty big one, water. But those molecules end up building cells. So a whole bunch of molecules come together to make phospholipids. Phospholipids make cell membrane to make cells. I kind of break this next part down. Another level that biologists study is cells make tissues, tissues make an organ, and then organs come together to make organ systems. So it would be like a heart cell making heart tissue, a whole bunch of heart tissue come together to make a heart, and then the cardiopulmonary system would come together to be obviously a system. All those organ systems come together to make an organism. There are biologists that will study just specific and 1 or 2 organisms. 2 or more organisms in a defined area is a population. So like Canis lupus, which are wolves, which tend to be kind of a popular topic in Idaho. That would be the study, some biologists will just study the population of wolves in Idaho. 2 or more populations living together in a defined area is a community. A community, you could have like the wolf, the tree, trees, grasses, elk, birds, and all those things, how they interact with each other, bacteria and other things like that. The communities, all the living plus the non-living matter, are ecosystems. The best one I can give you is a fish tank. Fish tank, you can't have really a fish tank without water. That's a non-living factor. You've got the interaction between that water and fish. You have air, CO2, and oxygen in there. So ecosystem is a living plus a non-living, and then all of the living places on the planet, or all the ecosystems put together, is what's called the biosphere. And there are actually people or scientists and biologists that look at the entire biosphere. So that is Unit 1.